This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 182, recorded on May 4th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How you doing today, Alan?
Doing well. Happy Star Wars Day.
What's that mean?
Star Wars Day. May the 4th be with you.
Oh, my gosh. Did you make that up? Or
No, that one's been kind of circulating around.
It's a meme, huh?
Yes.
How's the weather up there?
It's good. It's just finally cleared up after a long stretch of rain that we needed.
So your Skype thing, it says science, journalism, and everything in between.
Yeah.
Is there anything in between those two things?
I don't know, but I'd cover it if there were.
All right. You're just keeping your options open.
That's right.
Also joining us today from southeastern USA, Rich Condit.
Hi, fellas.
How you doing there?
I'm doing good.
I just had to say southeastern just to do something different.
Yeah, it's okay. You know,
You're
as
not
long as you get it right.
You're not insulted, right?
Not in the slightest. It's
All
okay.
right.
I never would have thought that I would end up here.
Well, you've been there a long time now.
I've been here for a long time, but, time flies.
And you're going to stay there, right? That's it.
I ain't leaving.
It's your final resting place?
Of course, never say never, right? But I got no plans to leave.
Rich, if they said we're going to establish a colony on the moon and grow pox viruses there, you'd go. Don't you think?
You think?
Yeah, they say we need a pox virus expert.
I have always wanted to go into space.
The object, and they're going to say, we want to see if you can do a plaque assay in...
Any excuse to go to the moon, I'm there. Right.
Here's a question for you, Rich. Do you think plaque assays work in zero gravity?
It's going to be tough. It's going to be really tough because you got to get some way for the fluid to stick to the cells to do the adsorption.
Right.
Once you get the adsorption done, you're okay. You
Yeah.
made it a centrifuge to do that.
That's true. Yeah, the overlay has to stay down. Good question. Not worth the billions of dollars it would cost to find out, though. We have a special guest today, our second guest in a row from the University of Michigan in Ann Arbor. Same department, microbiology and immunology, Mike Imperiale. Welcome, Mike.
Thanks for having me. It's great to be here.
Pleasure. Rich and I, of course, spoke with you way back in November and when we visited, and I think you were in the audience for the TWiV.
Yes, I was. I enjoyed it. That was great. I'm really glad you guys were able to make it out here.
Yeah, it's always hard to do these two visits. It's because we're trying to coordinate a couple of others, and it's not so easy. So anyway, that was fun. You guys are
That was good fun.
very welcoming, and you got great virology out there.
Indeed.
So you've been at Michigan a long time, right, Mike?
Almost 28 years.
Wow. It's 2 years shorter than me. Was that your first faculty position?
Yes, it was.
It
was my first time I lived west of the Hudson River.
Are you a New Yorker?
Yeah, I grew up in Brooklyn.
I guess I should know from your name.
So you lived that classic New Yorker cartoon, right? With the Hudson River and then flyover country in Los Angeles on the other side?
Absolutely. That was it.
It's amazing. So many scientists are from Brooklyn. It's really cool. So you were born in Brooklyn. Did you go to school in the city?
I went to high school in Brooklyn.
Okay.
And then I went to both college and graduate school at Columbia. And then I moved to the east side to the Rockefeller University for my postdoc.
Wow. Well, who did you do your PhD with at Columbia?
I did my PhD I was on the main campus, so
Right.
I wasn't up at PNS. And it was with a guy named Maurice Zauderer, who was an immunologist.
Wow.
Who was there at the time.
That's where I teach an undergrad virology course down there, so I'm there twice a week.
Mm-hmm. Yeah, we had to come up to PNS to take virology and immunology.
Cool. And a postdoc, who was that with?
Joe Nevins.
Oh.
Oh, no kidding.
You didn't know that, Rich.
And that was at the Rock?
Yeah. Mm-hmm.
I'm sure we went over this before, but,
he was
I'm
with Jim
guy.
Darnell's group at that time.
Yeah.
So when did polyomaviruses happen?
polyomaviruses happened when I came to Michigan. So one of the people who recruited me here was a scientist named Bill Brockman, who had trained with Dan Nathan at Hopkins. And he had been working with polyomaviruses, and unfortunately he passed away just before I arrived on campus. And the department asked me if I would take over as PI of his grant just to let it, finish out, let his students complete their dissertations. And I just became really interested in them, and so I kept the project going.
And 28 years later, here you are.
Here I am.
So, Brockman and Nathans, they developed BSC 40s, the cells I work with all the time. My mainstay cell line.
Yeah, there you go.
There you go.
Small world, huh?
Mike, someday we should have you on to talk about your polyoma work.
Yes, indeed.
Definitely. But today we are not. We're here because, among other things that you do, you're a member of the National Science Advisory Board for Biosecurity, the famous NSABB. How long have you been on that?
I've been on that since its inception. So our first meeting was in June of 2005.
Wow. I thought it had 7-year term. Oh, it must be you must be ending your term now, right?
Well, actually, my original letter of appointment says I had a 4-year term.
It's kind of like finishing out the grant, yeah.
Right. Wow. No, it's a no-cost extension.
Yeah, I was going to say no-cost extension.
So you were approached by someone to invite you on. How does that happen?
So, I think it was just serendipitous. So in the beginning of 2004, I was both chair of our institutional biosafety committee, and also at that time I was interim chair of our department because the dean was searching for a new chair. And there's this thing called the Association of Microbiology and Immunology Medical School Chairs or something like that.
Yes, I'm very familiar with this organization.
And they have this annual meeting, and that year it happened.
They have this annual meeting at fantastic places.
Exactly. And so that year it was in Hawaii. Right. And so I said, I'm not passing this up. So I went to this meeting in Hawaii, and one of the speakers who was there was Ron Atlas. And Ron had just finished serving on the National Research Council Committee, the so-called FINC committee,
right,
that had published this report, Biotechnology Research in an Age of Terrorism. And he spoke to us about, the committee's findings. And I thought, wow, it'd be really interesting to invite Ron up to Michigan to talk about this stuff, because I think people would be really interested in this. And then when I was talking to Ron, I came to learn that his daughter was a student at the law school here, so there was an even better reason to invite him up here. So when I got back, I went over to the vice president for research, and I said, I'd like to invite this guy. Is there any way we can find some money? And he said, let's put together a little symposium. And so we put together this little symposium, and one of the other guests who we invited was the first director of NSABB, a guy named Tom Holohan. And so he came and Ron, and then I can't remember who the other speakers were at this time. So I think there was just this sort of, confluence of events that led to me being on the radar there in Washington. And that's how I got asked to be on the board.
So you're not certain that your term is over yet. You're going to go on, I presume.
We've been told that until they find a replacement, we will remain on the board, and I don't know when they're going to find a replacement. So no one's asked for my opinion on anybody. So you know how these government committees are, right? They have to have a certain number of males, females, scientists, this part of the country, that part of the country. So
just like a study section.
Exactly.
Right?
Exactly.
I noticed that there's actually not a whole lot of females. Looks like about 20%, maybe.
Yeah, something like that, maybe.
That's it. That's an ongoing problem with pretty much all science committees, I think.
Right.
It's a problem with TWiV.
Yes.
Yeah.
Yeah. We try and we got to get 20. What do you need? 50%? Is that the
I
ideal?
assume they'd like 50%, right?
Sure.
Well, we got to have. We have to get 3 more members of TWiV, and all women.
My understanding is that in the wake of the genocide in Rwanda, their new constitution, by law, has to have at least 50% of the parliament female.
Uh-huh.
And it has become one of the most progressive African governments, if not progressive governments, around. partly, well, there's a correlation. We have to get an epidemiologist to study it, but I can't believe that wouldn't have an effect.
Right. So, Mike, tell us in a summary way what's the charge of the NSABB.
So, the NSABB is really charged with advising the government on issues relating to the dual use dilemma. And so there have been a number of different things that we've done in the years that we've been together. Probably the most important of which I'd say are we developed what we call the oversight framework, which really defines the issue. It's where we've discussed the whole concept of dual use and where we came to a definition, which you've probably seen, and I can read it to you if you're interested, where we decided that there had to be a threshold, right? Because almost anything can be misused, right? PCR can be misused. And obviously, we didn't want to be in a situation where we were putting impediments in the way of research. And that was one of our main guiding principles, was that we wanted to have a very, very tight threshold for experiments or types of research that might require a second look. And so this oversight framework discusses the discussions that went into that, the ideas, and then we have this definition. We took the 7 categories of experiments that were proposed by the NRC, by the FINC committee, and we expanded on those a little bit, and we listed those in there as the types of experiments that would be the most likely to fall under what we call dual-use research of concern. And then the other part of that document is a tool for how one might want to communicate research results that may fall into this category. And in fact, it's that tool that we used as we were going through the H5N1 papers. And so that was one big thing. And the other big thing that I think we've done that's really important is that we've discussed the issue of personnel reliability a lot. And this fell out of the charge that the FBI made that it was a U.S. scientist working in a United States Defense Department laboratory, Bruce Ivins, who sent the anthrax spores in the mail. And so the government then became very worried about what they call the insider threat and charged us with discussing whether or not there should be specific rules for who might have access to select agents. And we did a lot of talking to different people who are involved with personnel issues from various different public and private enterprises, legal people, ethicists. And we also came out with a report about that where really we recommended against any kind of draconian things. So some of the things that were being proposed was having people undergo credit checks before they could work in a lab and things like that. And again, we were trying to not put impediments in the way of getting the research done, because we thought that would be the biggest threat to national security, would be to not be doing the research. And then the other point I would just make is that one thing that people tend to forget when they discuss dual-use research is that just because something is tagged as having dual-use potential doesn't mean the work should not be done. It's more of a question of what happens after the work is done, and how do you deal with the results of that work.
Well, and hopefully what conditions should that work be done under?
Yeah.
Well, that's more of a biosafety issue, right, than a biosecurity issue oftentimes, because the biosecurity issues are covered mainly by the select agent legislation.
But, I mean, one of my concerns with this is, and certainly the way the process went down with the H5 papers, if you're stepping into research that was done in completely unclassified facilities and submitted through unclassified, weakly guarded peer review systems, and then coming in at the end and saying, oh, we're going to, try and control the information at this point, it seems like you're closing the barn door after the horses are out.
Oh, right. Sorry, I misunderstood what you were saying originally. Absolutely. So one of the things that we proposed, again, back in 2007, was that research should be examined throughout its lifespan, from conception to publication. So when you're thinking about the experiments, writing the grants, doing the experiments, analyzing the data, getting ready to present the work at a meeting, and then ultimately at the publication stage. So I absolutely agree with you that it would have been much better if some of these issues had been discussed more completely earlier on with respect to
which I think also kind of came out in the Fink report didn't it? That they had kind of this idea that there ought to be a more review of the stuff from the start.
Yes, exactly.
And my understanding is that process was never really implemented. Is that correct?
That process was not implemented. So towards the middle end of last year, the government started getting some input with respect to a policy that they had begun to draft that was based on our recommendations. And obviously that policy never came to fruition. Even the policy that they put out on March 29th is sort of a bare bones version of this more comprehensive policy that they were trying to develop.
Okay.
So recently, Health and Human Services issued a document which I think deals with some of this, right? They now say these pathogens, these kinds of experiments have to be reviewed before the work begins.
Right. So they now have, there as this was all going on, there was another panel which was called the Federal Experts Security Advisory Panel or something like that, FESAP. I know the acronym. And what they did, they were charged with looking at select agents and two things. Number one, are there things that should be removed off the list? Should the list be changed? And then they were also charged with looking at this issue of personnel reliability. And I think one of the most important recommendations that group made was to completely revamp the select agents list and to make what they call tier 1 agents, which they determined to be the most dangerous agents that we have to worry about. And there are a dozen of those. And so the government then added 3 other agents to that list, and that's what's in this policy that you were just talking about, Vincent. And basically what they're saying, if it's one of these agents and it fits one of those 7 categories, then that needs to be examined more closely throughout the process. And what Tony Fauci told us at the NSABB meeting at the end of March was that NIAID at least had done that, and they had identified around 10 projects, if I remember correctly, that sort of fell under that category and needed additional oversight, and I believe more than half of those involved influenza.
And they would be working with NSABB on this as well, right?
That's unclear to us at this point, as to how that's going to play out, whether we're going to be playing some sort of advisory role. It's unclear what role the institutions are going to be playing here. My personal preference would be that it would be much better if these things were handled at the local level and didn't have to get up. It's almost like recombinant DNA, right? It's better that local institutional biosafety committees are doing the bulk of the work, and only when you have special cases do you need a more centralized panel. In that case, it would be the RAC to deal with these things.
And my understanding is that that's what the Fink report originally proposed.
Right, sort of a RAC 2.0.
Yeah, and that's what we also proposed too. So we proposed a multi-step process where our proposal was that the PI is really in the best position to determine whether or not the work, is dual-use research of concern. And then if the PI thinks it may be a concern, then the institution should get involved, have some discussions. How can you assess the risks? What are the best ways to mitigate those risks? And then only if the institution were struggling with how to deal with it, either because of lack of expertise or whatever reason, then, maybe get NSABB or whatever other government agency, government advisory panel or the government itself involved. Because there's really, there's nothing specific in the NSABB charge right now that says we should be reviewing these types of experiments. Right? It's that would fall under a thing in the charge that says, and other things the government thinks is important.
So give us a sense of how NSABB reviews something. How does that process get initiated?
So for any of the topics that we're asked to come up with recommendations upon, what happens is that we usually put together a working group, which is a subset of the members. And the working group is constituted with mainly those people who are the most expert in that particular subject matter, and then other people who are interested. And then the working group will do the bulk of the work. Do the analysis, write an initial report, come up with the draft recommendations, and then those are given to the full board for additional discussion, and then the final recommendation gets made to the government.
And so how do you do the journals give you, for example, papers to review? Is that how it works? Because I know on the review forms there's a box that you can check, right?
Yeah. So as far as I know, the journals have never sent us anything directly, anything that we've ever received. And by anything, I mean the 1918 flu papers, and then these two papers have come from the government. So somehow the government has learned, that this work was out there. Well, with the 1918 papers, it was obvious because it was both came out of government labs.
Yeah.
But in this case here, I don't know. I've heard different stories, and it would just be speculation. My best guess is that I know, for example, when Nature reviews papers, they think it might pose some sort of danger. They do get a risk assessment from an independent person, and so maybe when they got that risk assessment, they decided, well, we need to let the government know or something. But I honestly don't know how those papers ended up on my desk.
Interesting, because I know there's on the review forms, at least for ASM. There's a box, I think, says, check this if there are experiments in this work of NSABB concern, something like that.
Right. And so what happens... Sorry, go ahead, Rich.
No. Go ahead.
I was going to say, what happens when you check that box is then the editor-in-chief of the journal has to look at it, and the editor-in-chief will often consult with Tom Schenck, who's the head of the publications committee or whatever it's called, and then come to a determination whether it should just go ahead or whether there is really an issue here that needs to be discussed at a higher level.
I see. But you say that
I don't think all journals do that.
That's right. Yeah.
Okay, I think JVI has been sort of a leader in this whole thing, among other reasons, because Lynn has been, still is, I guess, on the NSABB and is aware of the problem.
Right, and all the ASM journals do that. So I think it's, ASM took a very proactive role here.
Right.
It's interesting you say that you haven't received articles from the journals. You would think that would happen, but as you say, they come via other routes before the journals ever get a chance to do that, right?
I'm guessing, but I don't know.
Well, or the journals take it to the NIH, for example.
Yeah.
I mean, if it goes elsewhere in the government, it's going to get shunted ultimately to the NSABB, but not necessarily directly from the journal.
Yeah.
I would guess that any article that goes to NIH is going to ask NSABB to have at least a quick look at it, just to play it safe, just the way the government operates, right?
Yep.
Right.
But NSABB wasn't formulated specifically to be a watchdog over this kind of stuff, was it?
No, not at all.
I mean, there was supposed to be a process to take care of this, and NSABB, if I understand it correctly, was supposed to be advisory to the whole concept of dual use, and not necessarily a review board for papers.
Exactly. Our charge is to recommend policy.
Right. But in the absence of the other mechanisms, the responsibility falls to NSABB, kind of because you're the only guys in town.
Yep, I think that's the case.
So in the case of the Fouché and Kawaoka papers, they came to NSABB at the publication or review stage, but what we're saying is they should have been examined before that?
I think so. I mean, I think, certainly Ron has stated publicly that they had these discussions, at Erasmus Medical Center, and they determined that, there were not issues of concern. Ron himself was involved in similar types of discussion as NSABB in the Netherlands, and so he was well aware, I think, of these kinds of issues. I don't know what happened at Wisconsin, although my understanding is that they actually have a task force that deals with potential biosecurity issues, and that task force also looked at these. I think Yoshi mentioned something about that at one of the presentations he made. I can't remember which one now. Some of them are blurring in my head. So other groups did look at this somewhere along the line. Okay. But I don't know if anyone, for example, looked at the final data before it got written up and submitted for publication, other than the authors.
Right. Anyway, in any case, sometime last year, these two papers from Fouché and Kawaoka dealing with transmission of H5N1 influenza virus in ferrets ended up at NSABB, and the initial recommendation was to redact them in some way, to remove data or methods. And just this week, one of the two papers was now published because that initial decision was reversed, and we have the Kawaoka paper. It was published in Nature. It's called Experimental Adaptation of an Influenza H5HA Confers Respiratory Droplet Transmission to a Reassortant H5HA/H1N1 virus in ferrets. So I thought we would go over these results. I read them on Wednesday. It's really quite a nice paper.
Yeah.
And then then relate it to NSABB from there. So I think, as everyone knows who's listened to TWiV, the avian influenza viruses have a different receptor preference compared with human influenza viruses. They all bind sialic acids, but the chemical linkage of the sialic acid to the next sugar is different. The avian viruses prefer alpha-2,3 and the human alpha-2,6.
Oh, you've got that backwards, I think, Vincent.
Human alpha-2...
Humans alpha-2,6.
Yeah.
Yes, human
alpha-2,6, bird alpha-2,3. I know it's at one point was wrong in our textbook, but I've tried to learn it since then. So I remember a long time ago people saying that in order for these viruses to infect people, they have to switch receptor specificity, and of course they have to do other things as well. So this is a really neat study where they took the H5HA of a I think it's a Vietnamese isolate, and they basically mutate we know where the sialic acid binding site is because it's been studied many years ago, and they make random mutagenesis of the sialic acid receptor binding site. A couple of interesting technical aspects which I thought are neat. First, they replaced the multi-basic cleavage site in the HA, and that lets them do the experiments in BSL-2. So I guess if you have a plasmid just encoding the HA, Mike, if it's got the multi-basic cleavage site, you can't even work with that at BSL-2. Is that right?
I know that if you're working with highly pathogenic avian influenza, it's supposed to be BSL-3, but I'm not exactly sure if one has a reassortant.
Yeah.
As is the case like that. But again, no, I guess it must be BSL-3 because when they rebuilt that HA into the pathogenic H1N1 background, then they stepped it back up to BSL-3.
Right. Because they want to have if they want to go into animals, they want to have the multi-basic site.
Yeah, so initially they replaced the sequence so that they could work with it at BSL-2, and then for the later experiments, they go back. And
so I guess to do the mutagenesis, just selecting the viruses and so forth, you do that at BSL-2. It just makes it easier.
Right.
Exactly, yeah.
So the multi-basic site is, of course, what allows these HAs to be cleaved, which is needed for infectivity, by ubiquitous proteases that are in lots of tissues, and that may make these avian viruses more pathogenic and have a greater or a wider tropism. But they do random mutagenesis on the HA just around the cleavage site. They build viruses with the mutated HAs. They put it into a PR1, which is a 1934 influenza virus. And then they say, let's select for viruses from this library that can bind alpha-2,6, because we know the wild-type H5 doesn't like to bind alpha-2,6. So they take turkey erythrocytes, or turkey red blood cells. They treat them with an enzyme that takes off the alpha-2,3, because it's a bird erythrocyte. So now you're left with alpha-2,6, and they use these to bind the viruses and to select basically for those that bind alpha-2,6. It's cool.
Very similar to what was done in the Ruben Donis lab virology paper. Except they didn't use turkey erythrocytes. They had some other sort of solid phase presentation specifically of alpha-2,6 sialoglycans.
Yeah, that's right. They used...
and as a matter of fact, that becomes an interesting thing because I think the Donis lab paper used a pure reagent, whereas these turkey erythrocytes have lots of stuff on them.
Sure.
and the selection, therefore, I don't think is as tight as the Donis lab, because they came up with some stuff that on subsequent analysis was not really specific for alpha 2.6.
That's right. So they end up looking at, I think they initially screen 2 million viruses. They end up with 370 viruses after pulling them out with these turkey red blood cells. And in the end, 9 of them will bind to alpha 2.6 sialic acid containing red blood cells. All right. So they end up with 9 viruses. And they sequence those and they say, okay, they have various mutations in the receptor binding site. Some of them have been known before and some of them are new. So that's good. You get known ones, which means it validates your approach, and then you get new ones. You always want to get new things, otherwise you feel you're wasting your time.
Yeah.
And they have a wonderful figure in this. They have a wonderful figure in this paper that they keep referring to of the receptor binding site on the HA and the whole HA molecule that maps all these mutations and shows how it binds, and so it makes sense out of it.
Well, figure 1, it's very nice.
Very nice.
That actually helps a lot, because throughout this paper you're dealing with these strain names that's N158D, Q226, yada yada yada, and that's distinguished from one that's got an equally long name. So,
and I have to take a little sidetrack here. When I was first reading this paper, I'm looking at this figure, which is really clear, and I'm looking, how did they make this? It's made with MacPymol.
Yeah, I noticed that.
So I'm trying to make figures for a fields chapter, and I'm saying, what can I use to do this easily? I don't want to spend months and months learning command line structure programs. So I go to MacPymol after reading this paper. It's as easy as anything.
As easy as Pymol.
I set you up.
Yes. You just gave me the spike there.
I'm the straight man. I'm Costello, right?
Right.
I want to know why your chapter hasn't been submitted yet, Vincent. We had to submit ours a long time ago.
Oh,
I wasn't going to say anything, right?
Hey, it's submitted. I just wanted to fix some of the figures. But I have a few that I'm waiting for still. Well, the moral is don't get it in on time. All right. So anyway, we have 9 viruses, and they have now mutations, amino acid changes, some known, some new. And they do some more characterization of binding using now solid phase assays where they have defined sialic acids on plastic, and they can characterize the binding, especially comparing alpha 2.6 to alpha 2.3. And of all the mutants, only one likes 2.6 but doesn't like 2.3. So only one has become human-like in its receptor interaction.
And that has, 4 changes?
Yeah, that had 4. That had, well, I don't know if we should say them because it'll just confuse people. E119G, V152I, N224K, and Q226L. So I thought that was interesting. It's not so easy to go from a avian to a human receptor specificity, right?
No. That's the same lesson from the Donis paper, and I think here again, the use of the turkey erythrocytes rather than a pure reagent probably complicates this a little bit. If you're selecting with something that's a little more stringent, it might be a little more straightforward. But one of the things about that is that that's information in itself. It says that there's more to HA binding to a surface than just this receptor.
Right. That's right. The next thing they do is cool. They say, okay, we've made these viruses, we've characterized binding to sugars, but let's see what they do on cells. So they have human primary cultures, tracheal and lung cells. I guess they are.
These don't look like primary cultures. These are
tissue slices.
Tissue slices. Yeah, so they combine the virus and then detect the virus. I guess it's green fluorescence they're detecting it by.
Right. And by the way, we should mention this paper is free online.
Mm-hmm. Yeah, I noticed that. It's great.
They often do that with
important stuff,
particularly controversial stuff. So anybody who's listening to this who's trying to figure out these figures we're talking about, just pop on over to Nature and read it.
We'll have a link in the show notes that you can do. So we have tracheal tissues and lung tissues. All right. So tracheal farther up in the tract, and lung, of course, down lower. And because we do this because we know the alpha 2,3 and alpha 2,6 distribution is different. So in humans, you get alpha 2,6 sialic acids throughout the upper and lower tract, whereas the alpha 2,3 tend to be lung, lower tract. So they look at the binding of these viruses. So, of course, the parental H5 doesn't bind to the tracheal sections, but they bind to the lower tract. Let's see. The PR8 bound to both, so that's the human virus. Oh, the H5 PR8 recombinant bound poorly to the tracheal sections, but they all bind pretty well to the lung surface. And of course, down there you have alpha 2,3 and alpha 2,6 sialic acid receptors. Then the mutant viruses, so they have a collection of mutants with various HA changes. Some of them can bind to the tracheal cells, and some can't. All right, so the binding to the tracheal cells would be those that can interact with alpha 2,6 sialic acids. So not all of them have acquired the ability to do that, even though, again, in solid phase, they can bind alpha 2,6. In the actual lung, it's different. So you have some that don't and some that do. All right.
Right.
Anything else there?
Nope.
Okay.
Nope.
All right, so then they want to look at these in ferrets. And what they do is they put the H5s into a 2009 H1N1 virus. This is the new pandemic strain. So this virus now has all the genes from that 2009 virus and then just the H5 from this experimental manipulations that they've done.
And that to me is a critical aspect of all this.
Yes.
They're not looking at H5N1 virus. They're looking at the H5 hemagglutinin in a background that otherwise is entirely from the 2009 pandemic strain, which starts out, that strain is transmissible in ferrets.
Yeah, that does pretty well in ferrets. It's just the HA of H5 that we're looking at here. And every other gene is human, so it's a human viral gene.
Well, except that, remember, the H1N1 is a reassortant, really. But
yeah, it's true, yeah. It's from pigs and
birds. It's a mess. It's a mutt.
Mix. Right. But of course,
this is a virus that we know can transmit in ferrets and can transmit in humans, and there's no issue with its transmissibility, and we actually also know quite a lot about its pathogenesis thanks to the pandemic.
Right.
So they inoculate these ferrets with either. So you have all reassortant viruses. One is with the wild type H5, and then there's just an H1N1 alone, I believe they use, and then all of their various mutants. I think they look at two. They have 119, 152, 224, 226, and then 224, 226 alone. So a 4 amino acid change and a 2 amino acid change in this background of H1N1. And what they say is these viruses, these HA mutants, don't replicate well in the trachea. They're severely limited in their replicative ability in the trachea. Whereas and of course the H5 reassortant also doesn't replicate well, but the H1N1 alone replicates pretty well. And then they recover some viruses from these animals. So in other words, adapting these H5 HAs to recognize alpha 2,6 is not enough to get them to grow well in the ferret respiratory tract.
Right.
But they start to recover viruses from these animals, and they sequence them, and they find other changes that appear to help the replication of these viruses.
So hang on. These are intranasal inoculations, is that right?
That's right.
So that must, at least to some extent, bypass the trachea and go right down into the lungs. Because it didn't make any sense to me that you've got this, just the H5 virus itself doesn't bind to, according to the previous figures, doesn't bind to the trachea, doesn't bind alpha 2,6.
Right.
And yet, if you do an intranasal inoculation in ferrets, it replicates pretty well. So it must be, some of that at least must be going straight to the lungs.
Yeah, so they actually talk about that. I've lost my place here. So these viruses. It's funny. The H5 does replicate.
They get virus out of the trachea
in the turbinates.
Right. Yeah, but they also it does less well than the 2009 pandemic strain in the trachea by about a log, but it's still better than almost all of the other guys.
Yeah, so they don't understand that, I think.
Right.
But the point is, you make these mutations which change the specificity, and it gets worse in terms of replication. They add this one mutation that they find in one of the ferrets. It's a change at 158 asparagine to aspartic acid.
Right. So they've now passaged the virus in the ferrets, and they got out another change that they're trying.
Yeah. And this helps it replicate better. And they say this is a known amino acid change. It actually prevents glycosylation at this site, at this asparagine, and improves receptor binding. So you get a big sugar out of the way, and the virus binds better to its receptor. So this has previously been known. So this improves the replication in the ferret. All right. Okay so far?
Right.
Yep.
We're good.
Then they put these in ferrets and do a transmission experiment where you infect the ferret and then you have a cage next to it. And you ask if the virus can go through the air. No contact between the ferrets. This is all done in BSL-3+, which is the requirement, right, Mike, for these kinds of experiments?
I think the requirement is BSL-3, but I think that these guys used BSL-3+. My guess is just because they didn't know what to expect.
Okay.
Right. Makes sense.
Yeah, my sense is that Yoshi in particular was very careful with respect to the biosafety considerations of his work. You know, the whole idea, as we just discussed, of initially doing the experiments in a situation where you could use a lower level containment. I think he was, and also the fact that he did use this reassortant virus rather than just working with a straight H5N1 strain. I think he was concerned about, biosafety issues.
So in these transmission experiments, so if you just have H5HA, the wild type, in the H1N1 background, this is not transmitted among ferrets. The other viruses, 158, 224, 226, this one is transmitted. The 2-amino acid one, 224, 226, is not transmitted. So you got to add this 158, which they picked up in a ferret. You have 3 amino acid changes. This gets transmitted. It gets transmitted to 2 out of 6 ferrets.
Yeah, it's to me it's significant that the pandemic strain is they've got 3 different pairs they're testing, and it transmits within 3 days to all of the contacts.
Right.
None of these others are quite so transmissible.
Right. And they point out that...
Well, except that they do most of them seroconvert.
That's right.
Yes, okay.
That's right.
Good point.
Yeah, the seroconversion always seems to be higher than the virus recovery. So transmission is assessed by recovering virus from the animal, right? You do a nasal wash, and you see if you get virus out. And so that's not easy, because if you miss the peak of replication, you might not detect it, but seroconversion means the virus
I missed that. That's a good point.
Right.
So seroconversion is another story.
So seroconversion in 5 animals out of 6, as opposed to viruses recovered from 2 out of 6 with this triple mutant.
And none of these animals died.
Right, none of the animals
Correct.
died. Then they again recover virus from these animals that have acquired infection by airborne transmission. They sequence them, and they find another mutation. And this one is quite interesting because it's way far away from the receptor binding site. It's a threonine to isoleucine change at amino acid 318.
Right.
And this is way at the bottom of the HA near the fusion peptide. And it turns out if you put this mutation in now with the other 3, so that gives us 4 total changes in the HA, 158, 224, 226, 318. Now this virus replicates really well in ferrets, and it transmits 4 out of 6 ferrets. They all become seropositive, and no animals die. I bet you know these numbers by heart, Mike, right?
Absolutely.
Actually, 4 out of 6 have virus in nasal wash. 6 out of 6 seroconvert.
Yeah.
Right.
that's
and,
right.
and notice that the other thing that was kind of striking to me with that particular virus, and I think this is a really interesting mutation, is that the titers were higher, right? Indicating that you were probably getting better replication in those contact ferrets.
Right. So these ferrets do not die. They do get lung lesions, so they get an infection that leads to lung lesions. They do some pathology to look at that, but they're all surviving.
Right. So then they look at what this mutation is doing.
And in my view, this is the most interesting part.
This is cool
of this
absolutely
paper, because because this 318 mutation is at the fusion is near the fusion peptide. They say, well, maybe it's affecting pH sensitivity. So these viruses, when they enter cells, the low pH of the endosome triggers the HA to change its folding and expose the fusion peptide, and that's how the virus RNA gets out. So they say maybe there's a difference in fusion. So they check the pH level at which fusion occurs in these viruses. So they do a fusion assay where they put virus on cells at different pHs, and you can see the fusion that's occurred among cells caused by the virus. So when you just add virus to cells, if it causes fusion, you'll get these bigger cells forming. They're called polykaryons. So the wild-type virus has a threshold for fusion, pH 5.7. The double and triple mutant, the pH fusion is over 5.9. So that means at a higher pH, those two, the double and the triple mutant, will fuse. And then the fourth mutation, 318, that reduces the pH threshold back to 5.5. So their idea is that, so the pH apparently of the respiratory tract is 5.5 to 6, something like that, 5.5 to 6.5. So they say these, when you put mutations in the HA that allow it to recognize alpha-2,6-sialic acids, you're raising the fusion threshold, and these viruses are going to fuse in the respiratory tract before they have a chance to infect cells.
So you're destabilizing the protein, essentially.
Yeah. They actually do some temperature stability experiments which parallel that. So you don't want the fusion to occur outside of the cell, because then the RNA just gets put into the medium and it's useless. You want it to happen in the cell. So the wild-type virus fuses at 5.5, and that limits it to inside the cell. So this is cool. This tells you that the stability of the HA is really important for transmission, which makes perfect sense, but you wouldn't know that unless you did the work.
Right.
So that's where I think we learn a lot. We have now a mechanistic basis for transmission. We can adapt the virus to be transmissible, and to do that, it needs to have a stable HA with respect to pH in the respiratory tract. And this gives you the opportunity now to go out and look at all kinds of human strains and say, is this an important parameter in general, I think.
Right. And also I think to me it highlights how inconsequential the specific mutations are. Because what we're looking at here is the mechanism behind them.
Right.
So it's not necessarily the case that you need these 4 mutations for an H5 virus to become transmissible in mammals. But we're looking at a pattern that you need to have. You need to change the receptor specificity by making these changes in the head of the protein. And you are also going to need to stabilize it after you've made those changes.
Right.
If the initial changes destabilize it, okay, which in this case they did.
They did. And that may or may not be a general phenomenon, but it's something that we certainly didn't suspect before.
Yeah. This graph in, figure 5A is really interesting because it shows the pH profile for the heterokaryon formation, and you can see that the wild type has a certain profile, and the initial mutants are shifted to a different profile, and the compensating, stabilizing mutation has a totally opposite profile, but the combination of them returns the profile of the wild-type profile. It's very interesting.
It's very cool. Now, so they point out in the discussion so they say, we don't know whether these mutations would allow transmission of this virus in people, which is fair because we don't know that. And they say, look, we have all H1N1 and just the HA from H5, so we know that other genes are probably important. And people have found, in fact, that polymerase genes, for example, are important for transmissibility. And so all they're looking at, actually, I think it's a nice approach. You just focus on HA and you say what's important there.
Right.
And it allows you to hone in on that. One other important thing I think is worth noting, 3 of these residues, 224, 226, and 318, they say have been strictly conserved among H5HAs isolated since 2003.
Yes.
So in other words, the mutations they see in these experiments, they haven't been found in nature. Is that right, Mike?
I don't remember exactly. At the NSABB meeting, we got presented with a manuscript in which they've analyzed a bunch of naturally occurring strains. Mm-hmm. And because we weren't able to keep notes, I don't remember exactly which mutations.
I see.
I think that there are viruses in nature, if I remember correctly, that are 2 mutations away from this virus, this 4 mutation virus. But I don't remember where those 2. So that may be the 224 and the 226.
Yeah.
But I don't remember exactly.
You can't take notes, huh?
Because at that time, at that time, both papers were under export control regulations. So the U.S. slapped an export control, consideration on Yoshi's paper, and then the Dutch government did it to Ron's paper.
After this stuff has already been emailed around the world a few times.
Well,
I know you do what you can.
Well, I can tell you the first time around, right back in the fall when we saw the original manuscripts, we were only given hard copies. But right, it was emailed or uploaded to the Science and Nature websites.
Yeah,
right?
exactly.
So anyway, we had to sign these legal documents basically saying, we were going to read the papers, we were going to then we weren't allowed to take notes, and then we would turn them in at the end of the day. And so that's where we were at. So if you I can't answer your question based on.
Yeah, I can understand. It's too bad because this stuff is so complicated. I have to take notes when I read.
Yeah,
papers,
what I did was I scribbled notes on the manuscript itself.
I see. Yeah.
Right.
So the initial papers that you guys saw, you decided should be redacted, and then not too long ago you changed your mind. So I don't know if you can give us some insight into that. Is this paper that you see here essentially, in terms of data, what you saw originally?
So this paper, in terms of data, is essentially the same. The 2 things have changed. 3 things have changed. First is some of the supplemental data and actual data was kind of swapped around and stuff. So, and I think that made it clearer in the actual paper without having to refer to all the supplemental data. Exactly what was going on here. So it just made the presentation clearer. Then the second thing that, if I remember correctly, and again, because I don't have a copy of the original paper in front of me, but I was paying attention to this when I was reading them back in Bethesda. The data in this paper with the single mutation at the threonine 318 was not in the original paper. So I think it does add, some really, as we just discussed, some very interesting points to the paper. And then the third thing that's different is just the way that the paper is presented, in terms of it making it very, very clear what can and cannot be concluded from this paper.
So what was the initial concern about this?
So the initial concern about this paper was that if someone were to take these exact mutations in the H5 H5HA and reconstruct that onto an authentic H5H1 background, that could be a very dangerous virus.
Or it could go nowhere.
Right? So we don't know. Okay. So the question is, what are the benefits and what are the risks? So the benefits, I think, are twofold. Well, besides let's make it a given, right, that the science is important. Okay?
Yeah.
I think we can all agree on that. I personally disagree with those who have gone out and said these experiments should never have been done. I think that's just wrong. So the benefits are, will this help public health surveillance? And will this help in terms of vaccine development? So, I think with respect to vaccine development, since really, especially the inactivated vaccine, the whole point of that vaccine is to induce a neutralizing antibody response, right? So whether or not these mutations, whether vaccines would have to be tested against this exact H5, that's not clear to me. So that's one thing. And then with respect to the public health surveillance, so, Alan, I think you raised a really good point just a few minutes ago that what's really important here is the phenotype, right? Not so much the genotype.
Right.
And so this would help public health surveillance to some degree because they could start looking for these particular mutations, but there are probably a lot of other ways to get to transmissibility. Right. So in that way, the utility to public health surveillance was somewhat limited.
But I think we shouldn't focus narrowly on immediate applications of a basic research paper.
No, I agree with you completely.
Because, the mechanistic insight that you get here is actually quite significant, I think.
I agree. I'm just giving you our rationale, okay?
Sure, okay.
Talk about the bigger issues because I don't disagree with you there. And then the other issue that with respect to public health surveillance and just the nature of influenza is that because of the way the system is set up, by the time the mutations were identified, it's pretty likely that the virus will have already started to spread. And so then it becomes a containment issue. So there were benefits, but there are some caveats to those benefits. Now, what about the risks? So the risks are, would someone be able to either reconstruct this virus, right, because they want to do harm for whatever reason, number one, or number two, is it possible that this virus might be accidentally released from a laboratory? So with respect to intentional misuse, it's pretty obvious, and it was obvious to us back in October or November when we first reviewed these manuscripts, that the methods that were used to generate this virus are out there. Anyone could go into the literature and know, what, all we have to do is really, if you think about it, mutate the receptor specificity. So get that alpha 2,6 specificity into the HA and then passage it through ferrets, right? And we could get something out that's virulent.
Well, you could get something out that's this virus.
Transmissible.
That may be transmissible between ferrets.
Or it could be a different virus. Remember, these guys only started with a pool of 2 times 10 to the 6th.
Right.
And got 1. But anyway, the thing is that the question, so what it came down to was, okay, can we at least slow someone who's intent on doing harm, slow them down by not giving them the exact mutations, all right? So that's the intentional misuse part of it. And then the unintentional release thing is that, look, we all know that BSL-3 laboratories have accidents all the time, right? And so there's the potential then that this might accidentally be spread. And we know that there are antiviral drugs available, sure, but we know there have been cases, for example, with brucella, where individuals have become infected in a BSL-3 lab, and they didn't realize it, and they get sick, and I think there was a case just recently in Chicago, right, where an individual died. So we know that accidental release happens. Okay, so now how do you weigh those risks and benefits? The utility with respect to public health, and then the possible risks. And so I think that our original recommendation really struck a good balance between those two things, because we realized that we could mitigate the potential risk by, again, not giving someone an exact template on how to engineer transmissibility into this virus, but at the same time, by providing the information to those who need to know, public health officials, other scientists who have a legitimate reason to know these data and move forward with the types of experiments that need to be done. We wanted those individuals and those public health systems to know this information. And so that's why we recommended redaction, but let's be able to share that information with those who need to know.
And did you detail a method by which that information would be shared and how you would clear people to access it?
So we did not detail a mechanism. We were made to understand by the U.S. government that such a mechanism could be put in place. Okay, and so do you want me to continue, Vincent, with what happened the second time around? Then?
Sure.
Because I think
Yeah.
okay. So here's the key, right? Because now we get brought back, to the NIH. Well, I shouldn't say brought back. The first review was all done by teleconference.
Right.
We get brought to the NIH at the end of March, and at that meeting, we're told, "Hey, there is no way to share this information, only with those who need to know."
Is that right? So that's why you were brought back?
Well, I mean, that was part of it. The other reason we were brought back was that the authors felt that we had misunderstood
I see.
the original manuscripts.
And also the WHO reached a different conclusion.
And the WHO reached a different conclusion. But I think it's not surprising that the WHO reached a different conclusion, since that was a room full of influenza experts.
Well, and it's also, I think a lot of people don't quite appreciate how much of a divide there is between the U.S. and most of the rest of the world on this perception of biodefense and biosecurity. There are very few other countries that take this quite as seriously as we do.
Absolutely. And that's another thing that at least played into my decision-making the second time around.
And by the way, I'm not saying that the U.S. approach is correct on that either.
No, I understand. So anyway, we were brought back and we were told that there were two reasons that you could do limited sharing. The first reason being this issue of export controls. So, for example, let's assume that the U.S. government were the repository for this information. Well, then anytime someone in another country wanted this information, then the U.S. would have to issue an export license.
Right. It's
not an easy procedure to go through. It'd be very tedious. And then on the other side of the coin, we were told that anybody who wanted information, access to this information from within the U.S. could put in a Freedom of Information Act request. Now, a number of us asked, wait a minute, can't the U.S. government deny this request given the nature of the information? And Francis Collins told us that their lawyers said they would not be able to defend that in court.
Wow.
Now, I actually hypothesized this very early on.
But here's the funny thing, Alan, is that I as you might imagine, I've been speaking to a lot of reporters over the past few months, and I've heard from a number of reporters that they've put in FOIA requests for much more innocuous things than this and been denied.
Well, so, the frontline response is often a denial, but if you appeal it, you usually get through, and there's a whole negotiation process that goes into a FOIA request. Right. So yes, the government could stall, right. But it seems to me that with federally funded research done in an unclassified setting like this, it would be virtually impossible for them to stall indefinitely.
Exactly. So what it came down to me, guys, is that in my view, the risks didn't change, the benefits didn't change, but what changed is how are we going to be able to realize those benefits? Okay? And right. So because of these potential roadblocks in terms of implementing what we had originally recommended, my feeling was that the only way we're going to be able to realize these benefits was to allow for publication of all the information. And so that's what drove my vote the second time around.
So the NSABB, it was convened as a committee to make policy recommendations, suddenly had these two papers dropped in your lap, which was not really supposed to be your mission to begin with. And I guess under the circumstances, you came up with a solution that seemed to make sense, but you didn't have the information to know whether or not that solution would make sense.
Right. So the thing, and speaking of not making sense, is that, when we had all these discussions back in the fall, there were many of the ex officio members. So the ex officio members of NSABB represent all the government agencies
right.
that, have an quote unquote interest in life science research. And not a single one of them ever said, hey, wait a minute, we better think about export controls, which is mind boggling to me, because this could have been avoided. Although, to be honest with you, back then, based on the way the papers were written, who knows? We may have recommended don't publish at all. I'm not going to speculate on what our decision may have been back then. If they had told us back then, the only two options are publish in full or don't publish.
Mm-hmm.
Also, just a kind of a meta process observation. It's been my experience on conference calls that the more people you involve in the call, the less information actually gets transferred. So it may not have helped that all of this was being done remotely.
Yeah, I'm not a big fan of conference calls.
So basically, you think that the rewriting of the manuscript helped a lot, right?
I think the rewriting did help a lot, although again, I'm not sure it really changed what I view to be the risks.
Yeah. It's just the export issue that really clinched it in the end, right?
For me, yes.
Now, what would happen in a month if you got another set of papers on a similar topic? Would it revolve again around export issues?
Oh, that's a great question. I'm guessing that, since the government is now implementing this new policy, that something will happen before a paper gets submitted.
I would hope so.
I certainly hope so.
And then that's going to be maybe a negotiation between the government and the institution and the investigators as to.
My understanding is that in pursuit of this new policy, they're reviewing all of the current grants out there and identifying ones that might have potential for dual use, and going through the process that we should have been going through from the beginning.
Exactly, and there's no doubt this is going to come up again. There's absolutely no doubt.
Sure.
Yeah, sure.
Yeah.
I don't know if we can deal with this because the other paper's not out yet, but there was a difference in the vote. This time the vote was revealed, right? So the Kawaoka was a unanimous decision, and the Fouchier paper was 12-6. Is that something we can understand, or should we wait for the Fouchier paper?
No, here's how I think we can understand that, and that is that because the Fouchier paper had not yet, the revised Fouchier paper had not yet been submitted for publication, okay, there were still some unclear things in there. And my sense, and again, because as I just said, I voted for publication, and I can't speak for my colleagues, but my sense, based on this discussion, was that there was unease because of the remaining lack of clarity.
So you would have been right. You were
and
voting.
The way that so we were voting on a paper that hasn't been peer reviewed.
Right.
Okay. And so my understanding of the motion that was made was that after peer review, the paper didn't say what we were led to understand it was going to say, that then it shouldn't be published. In other words, our recommendation no longer holds. And we explained that very carefully to Bruce Alberts, who is the editor of Science. And so, I'm hoping that's going to play out. I'll be honest with you, if I read the Fouchier paper and it says something different from what I was led to believe, I'm going to be very outspoken about that.
I'm interested in how the NSABB assesses risk. To what extent do you hear testimony from various biodefense, biosecurity, bioterrorist type individuals?
Yeah, so the first time around, we did not receive any kind of classified briefing. So all of us on NSABB have at least a secret level security clearance. At the in-person meeting at the end of March, we did receive a security briefing. So that's how we hear about those types of risks. So, those are the sort of, if you will, concrete risks that the intelligence community knows about. But then there are the types of risks that, we've just been discussing over the past few minutes that I think anyone can glean just from knowing the nature of the beast.
Okay.
This is really something that we should probably explore another time, and we actually have
we have some correspondence that leads into that.
Because I think I find risk assessment, pretty murky.
I find it very difficult, and it's one of the things that when NSABB was first starting, I actually went into the literature and tried to, learn as much as I could about risk assessments, and in particular, in situations where it's hard to quantify that risk.
Yeah.
Right?
Yeah.
Right.
And it really is. It's very, 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 very difficult.
Right. I think it would be important to get somebody on the show who can speak to this.
Yeah. Well, maybe as we'll see in the correspondence, maybe we have someone. Mike, I wanted to ask you just a couple of other things about what you had said. So you mentioned allowing someone to take these mutations and put them in virus and then pass it in ferrets. Well, couldn't it be that someone would just pass it in people? If you have individuals who want to use this for nefarious purposes, they would certainly be able to recruit individuals, which would be far worse than anything we could imagine, right?
Unfortunately, I think you're right.
So that's not something that you could ever control.
Right, exactly.
You also said that...
So here's an important thing to remember, right, Vincent, is that when no one's ever going to stop someone who's really intent on doing harm from doing it, right? You can only lower the risk as much as possible, right? But if you're going to try to bring the risk to zero, then the corollary is you're going to make no progress.
Yeah, exactly.
Right? The only way to bring the risk to zero is to say we're not going to do this type of research, which, in my view, is not tenable.
Yeah, I mean, it really comes back to risk-benefit again. And my view has always been in science that sometimes you don't know the benefit, so it's really hard to do that equation.
Right.
And I would err on the side of assuming there's more benefit than you know, because just as you know, having seen how science works, there's so much serendipity.
Yeah, and as I said, the last thing we want to do is put an impediment in the way of science. Yeah. So you can imagine how afraid we were, so to speak, of this particular risk to come to this decision.
Well, I think it's important to note that, as you say, the NSABB members are interested in science going forward, because the climate that we got from the NSABB through the press was that you are draconian guys who are going to just shut this down and we don't care about anything else. But you can deny that if you'd like.
No, and actually it's even better than that, Vincent, because it's not just the scientists on NSABB. It's the non-scientists. It's the people from the intelligence community. They want to see this stuff move forward because everybody understands that's our best defense, is being out ahead of the curve.
Right. I think the intelligence community may also have a fairly realistic understanding of the history of biodefense. I mean, we actually have examples of non-state groups developing and deploying biological weapons, and they have it's just ridiculous failure after failure.
Right.
I hope they do.
I mean, it doesn't work. It's a horrible place to invest your resources if you're a terrorist group.
Right. I mean, what everybody always says is that the terrorist only has to get lucky once. We have to be prepared in a perfect manner all the time, right? So.
Well, isn't the best preparedness having vaccines and antivirals, right? Instead of worrying about what research gets published, why don't we work on, a universal flu vaccine or antivirals, stockpile antivirals?
Absolutely. But remember, up until recently, I think up until these papers were known, there was no impetus to make an H5N1 vaccine, or very little impetus to make an H5N1 vaccine, because people didn't think it was going to be able to become transmissible. So I think that's another reason why it was important that the results of these experiments be known.
Well, if that's the case, then the message is make a vaccine, make an antiviral, and let the work go forward.
I really hope we can come up with a universal vaccine. I think that would be wonderful, right? Because then that lets you move forward on all influenza research without these sorts of issues.
Right, and realistically, the much, much bigger threat to public health by many orders of magnitude is a naturally occurring virus.
Mm-hmm.
Absolutely. Mother Nature is the best terrorist, right?
Yep. Right.
All right, so another issue I wanted to address. You mentioned in your discussion a little while ago of the WHO meeting that, it's full of influenza scientists, so we're not surprised at that result. And this sort of ties into the Osterholm letter that I wanted to mention. So don't you think that, the influenza people would be getting the results anyway, even if it were not published? Don't you think that they're the best able to evaluate the risk of this? And so why would it be just because they're flu virologists that they're biased? And it's the same charge that Osterholm has.
Yeah, sorry, I didn't mean to imply that they were biased. I was just trying to make the distinction that it was two different groups of individuals looking at the papers.
So you don't think that inherently they're going to, ignore risk just to further their own research.
I don't think inherently. I think what I would argue, though, is that because the NSABB is just a little bit more broadly constituted, that we might just have a slightly different view of things.
Well, sure. You got all the intelligence people and the policy people, the response. You have lawyers, epidemiologists, so it's going to be different, of course.
Yeah, that's the only point I was trying to make.
Okay.
I wasn't trying to.
So Osterholm sent a letter to, I don't know, NIH. He's basically complained about this re-review of NSABB. And you had pointed out this to me a while ago when we were discussing your coming on here. You know, he said that the committee was set up or in a way to guarantee this outcome. And, you know, in particular, just putting flu people and excluding people who would talk about risk. So do you have any thoughts about that?
I could give you my personal thoughts. Obviously, I can't tell you whether the other 20, however, members were influenced, you know, in one way or another by the way that the agenda was set up. But, you know, guys, one of the things that I have done over the past 6 months is learned an incredible amount about influenza. So, you know, I've delved into the literature, you know, on hemagglutinin, transmissibility, receptor binding, RNA virus genetics, which, you know, I don't have to tell you guys what an amazingly complicated field that is. Learned as much as I could about, you know, public health, surveillance for influenza. So I felt that I walked into that meeting with a pretty good sense of, you know, where everything stood. It was pretty clear to me from statements that Tony Fauci made before the meeting even what he would have liked to see the outcome be. Mm-hmm. So I don't think the NIH hid anything in that regard. And during the meeting, Fauci even came out and said it, you know, that they'd really like to see these papers published. So now, you know, did I personally feel pressured to make a decision based on either his statement or the way the meeting was set up? So again, I felt that I had enough information to make a reasonable decision, number one. Number two, there were experts in the room who were not from the U.S. government and who were very helpful in answering at least some of my questions that were lingering. And finally, the thing that I would say is, you know, I don't answer to Tony Fauci. I don't answer to Francis Collins. I feel that my responsibility is to the public worldwide. What do I think is going to best benefit the public with respect to influenza? And so that's what drove my decision. You know, if I would have come to the decision that I needed to recommend against publication, I really would not have cared what Tony Fauci thinks about that or the NIH in general. So if I thought that was best for the public, then I think that's so I guess what I'm saying is that, you know, I think that the NIH was didn't hide any agenda. It was pretty clear what they would have liked to have seen done. How that influenced others on the board, if it did at all, I can't answer that.
So you don't agree with the tone of this letter, then?
I think he raises some legitimate points. I mean, if you just look at the way the you know, who did we heard from the authors? We heard about this third paper with this computational modeling of whether or not mutations out in the field, you know, may lead to these mutations and what the possibilities are. Honestly, I'll be honest with you guys, although I have great respect for the lead author on that paper, I didn't really give it a lot of weight because, number one, it was obviously biased because both Ron and Yoshi were co-authors on the paper. Number two, as far as I know, it hadn't undergone any peer review, so I don't know whether to believe any of that. And just my sense, again, from what I know about RNA virus genetics, was that it would be pretty tough to model what might or might not happen. So I really did not put a lot of weight on that paper.
Well, this letter, I guess, was supposed to be confidential, but it was leaked, and so it's out there.
Yeah, it's unfortunate. And you know, Amy Patterson did respond to Mike, and I guess that letter has not been leaked, I'm assuming. So, but you know, I think it was a very thoughtful response.
Yeah, well, but I don't think Mike's position on this was a secret at any point. He was pretty outspoken about a lot of this.
No, it's quite clear where he stands. He even says in this letter that he didn't support the 1918 H1N1 virus publication. It's in 25. I guess the NSABB reviewed that, right?
Yeah,
it's on the record. And he said that shouldn't have been released. But now in fact,
I think he
did support it
for
originally,
I think he said
but
if
he
he
says
had to
that was
vote
a
now, he
mistake.
would.
Oh, sorry, he made a
said that
mistake.
was a mistake.
Yeah, we made a mistake in approving publication. Well, now, in fact, if you got 2009 H1N1, you'd be immune to the 1918 virus, so that's not really an issue any longer.
Well, and in my view, the 1918 paper was different because the sequence of all the segments of that virus were already known.
Right.
And really what that group did, they took them all together and made a virus out of it. So the sequence was already known. How to make influenza virus from sequence was already known.
Yeah.
So those guys weren't really telling us anything new, you know?
Yeah, yeah. Okay. Anything else before we move on, Rich and Alan?
No.
I'm cool.
Got
I
everything
think we've covered this pretty well.
All right. So I think you've clarified a lot of things.
Definitely.
I appreciate that.
Great. I appreciate the opportunity because, as you said, you know, the way that NSABB is being perceived out there is a little bit unrealistic.
Yeah, and that's why I wanted you to come, because we get a lot of letters from people who say, well, I don't understand. Why did they change their mind? What's different? And
right.
I've talked to some other members, and they say, well, I can't talk because it's all confidential, and it doesn't help. But, you know.
Well, we can't talk about the details of Ron's paper, but, you know, the process and the thoughts and everything, I think, is fair game. And,
what you've done today
I hope this was useful to you and hopefully then to the listeners.
It's useful to us. I hope the listeners.
It's extremely useful to me.
You know, when the Fouché paper comes out, maybe we'll talk about that as well.
Yeah.
We had a little correspondence with someone sent us a link, I believe, to. Do you know Peter Sandman, Mike?
I did go to his link after, was it last week's podcast or the week before? I can't remember where.
Yeah.
I think it was Jesse Bloom who pointed that out.
That's right. And so we read Jesse Bloom's letter, and so Sandman is rather critical of both sides. He said, you know, the scientists haven't helped and the policy people haven't helped, and what we need is a dialogue. So we had a little response here from a listener, Joe, who has been listening for quite some time, and Joe is a person who is in the response field, and so Joe writes, he wrote a letter to Peter Sandman. Basically, he says, "Here's the text of my post. I was disappointed in the quality of his article, as I have much previous experience with his work, and see him as the David Baltimore of risk communication. If you could get him as a guest, you would enjoy the discussion. He reminds me of D3 in his ability to tell a story." And D3, of course, is Dixon Despommier. So he writes, "Peter, while I have the greatest respect for you and have been to trainings by yourself and Vincent Covello in years past," what a great name.
Yeah.
2 L's and an O. Wow.
And a Vincent.
And a Vincent. Has to be a good guy. I have to tell you that this article is very flawed, both in the facts used and in the conclusions. About 5 years ago, I did the work to prepare my global organization for responding to a potential pandemic based on my concerns over H5N1, SARS, and a few other pathogens. At the time, I did the work to get as complete an understanding of the state of the science that I could achieve with my background in chemical engineering and environmental science. Within a few years, the public discussion caught up with risk management trades discussion on pandemics, and we saw serious resources put into preparedness planning at the international, national, and local levels. We have relatively robust systems in place and are working to improve the major deficiencies in prediction, vaccination, and control. The research and the researchers you are unhappy with have been an integral part in trying to get the answers risk managers like me need to make good decisions related to these preparations. Their emotional response to the criticism of a faceless, unknown committee is very understandable to me. Their efforts to remain professional is to be praised, and their lack of sophistication regarding risk communication seems to be something they are working to rectify. So that's interesting, Mike. There you go, a faceless committee, and that's, I guess, part of the problem. So you're a face, so I'm glad you did this. I would argue that decide, announce, defend strategy that you describe better describes the NSABB's approach than the scientists, and the target of their decision was not a few papers, but the fundamental process of scientific communication among peers by publishing finds for peer review. Scientists value this in a way equivalent to the First Amendment of the U.S. Constitution. To me, their outrage is appropriate and reasonably targeted. With response to the facts, we know the 60% number is not accurate in the way you used it. This is the case fatality rate. It is the percent of those who sought hospital care that died. The number of people infected is certainly much larger, and there are good studies that indicate that this flu is currently in or near the typical flu case fatality range of 0.1 to 2%. This flu strain is not exceptional in its lethality as you characterize it. Ebola, Nipah, HIV, Hep B, SARS, and many other pathogens are very similar to it or worse than it in virulence. There is little mystery in how the 4 biosafety levels of protection are determined for a pathogen, and he lists them here, and we'll go through them. The BSL system has been incredibly successful at stopping lab-acquired infections and loss of containment, notwithstanding unproven speculation about the source of H1N1. Note that it has an excellent reservoir in pigs where it experiences little evolutionary pressure. These experiments are not as unusual as you suggest. The value in publishing them is in accelerating our ability to develop an effective treatment or vaccine, to build our defenses against a natural or unnatural outbreak. Their work is totally reproducible by any competent biologist with a supply of ferrets and virus, both readily available. My view is that the NSABB did not understand the history of research in this area, made a knee-jerk decision to ban the details, and were caught by surprise when knowledgeable people protested the impacts of their decision on all scientists. All right, so that's Joe's letter.
I'm curious, Mike, did you anticipate the flap that the initial decision would cause?
Sure. Mm-hmm.
I would imagine that if you tell people you're going to redact a publication, it would be a pretty strong response.
It was distasteful to me as a scientist,
right,
to make a decision like that. But I just would make 2 points about Joe's letter, and I think he makes some very good points. First of all, I completely disagree with his last sentence, and I hope I've convinced you over the past hour and 20 minutes why. But the second thing that I disagree with is that, I think scientific publication is not a First Amendment right, and that we do have a responsibility to not publish things that might put people at risk. That's our role as scientists. We have a responsibility to the public. Just as I said for myself throughout this process, that my responsibility is to the public, and that's because I am a scientist. I'm funded by the public. I'm doing the work I do to enhance public health. And so I think we have to be a little bit careful there. I think we can't hide behind First Amendment rights. I think we can't hide behind even issues of academic freedom.
I think we can't ignore First Amendment rights or academic freedom either.
I completely agree. But I think we have to be careful that we can't just put a blanket statement out there saying I could publish whatever I want.
Right, but the notion, I think the point that he was making, which is one, or at least this is what I read from it, is that it is quite a radical step for an advisory panel to say to the authors of the paper, you need to redact details of your research.
Absolutely.
That's a departure from quite a long tradition that has good reasons for being held.
Right, and with most of us on the committee being scientists, we were very well aware of that.
Right.
Let me tell you, that was not an easy decision to make. I can't tell you how many dozens of hours went into just the teleconferences, much less the amount of time any given individual may have spent outside of those conferences, reading the papers, reading other things. As I said, the types of research that I myself did was countless numbers of hours. And I was trying to do my day job at the same time.
Right.
So Peter responded to Joe, Peter Sandman, and said, okay, I'm going to draft a response to you, but in the meantime, here are some links to all the articles I've written, and he links them all. And then he says at the end of his email, I'm not sure whether it makes sense for me to participate in a future TWiV or not. I'd be interested if the focus were on risk communication issues raised by the controversy, not the technical issues where the moderators are experts and I'm a novice. Even if I were right, or more likely half right on the technical point, they'd still outrank me and could still out-argue me. But given that the papers are headed for publication now, a conversation about the risk communication implications of the mooted publication debate would probably seem pretty peripheral to the TWiV audience, I think. Still, I'd be open to an invitation of the 3 moderators if the 3 moderators think it makes sense. And I won't feel slighted if they think the moment has passed.
I think it makes sense.
I think it makes sense. I'm really interested in this whole issue of, I don't even know what risk communication is. And I'm interested in the whole issue. And this is not about having an argument. This is about, learning to understand each other.
This issue of risk communication, I mean, I've come across it in bits and pieces in my work, but this guy, this is what he does, and I think it would be incredibly useful to have him on the show and talk about, and not the technical issues of what the virus does, but just in general.
Yeah, I think I'd like to know how you make these risk assessments.
Yeah,
so
absolutely.
I'll email Peter and invite him on.
I think it would be great. In fact, I would make it part of a bigger discussion of what are we going to do in the future when these types of situations come up?
Yeah.
How are we going to deal with it? Because I think we'd all agree the current way was not the best way to deal with it.
Right.
No. Yeah, and as Joe's response letter indicates, this is not just about flu, and it's not just about dual-use research. There's a whole lot to do with risk communication in virology. And the whole vaccine issue that's been, unfortunately, a lot of public confusion over that over the past couple of decades. That's an issue of risk communication as well.
Yeah, good point.
So, Alan, why don't you read Joe's response?
Right. So Joe responds, Peter, it's interesting to me as we continue this dialogue, and now that I've read the actual paper discussed on TWiV, that it is a perfect example of how much people can disagree even when given the same facts. I don't think the work that has created the ruckus is that different than similar work going on all over the world with a variety of organisms, many that I find much more concerning than H5N1. I must admit that I don't watch TV news, so I may have missed some of the drama. My view at the time was that the researcher, Dr. Fouchier, created the problem by announcing to the media that he had created the world's scariest virus, assuming the facts I saw are accurate. I later heard him talk on the TWiV episode in Dublin, and he seemed much more circumspect now that he has experienced some live-fire media training. That's exactly my perception of it as well, by the way. Now that I know he had to pre-mutate the wild virus to give it a starting chance to passage through the ferrets, I find the risk of a pandemic lower, not higher than before. What he did actually prove just how high the natural barrier is between H5N1 and mammals. There's a lot of research being done in BSL 3 and 4 labs that could be much more worrisome than this if it was not done with the extreme caution and rigid rules of the BSL 3 and 4 labs. Since one of my jobs is to coordinate with the safety staff that run the BSL 3 and 4 labs in the USA to allow our field service engineers to come out and repair our instruments in their labs, I know just how seriously everyone takes these rules. When you say the virus is stored in a lab, that does not quite conjure up the reality of how they're stored in a double-sealed container in an isolation system within at least 2 other isolation systems. BSL 4 labs live within a BSL 3 lab that is usually attached to a BSL 2 lab. Has there been a lot of media coverage of angry scientists on this issue? I haven't seen it, but would not expect to if it does not show up on the web. I ask because I would not characterize most researchers in the biotech world as dismissive of public concerns. These are, in fact, the same folks who volunteered to put on the Asilomar conference when folks were scared of genetic engineering in the 80s. I can't think of many branches of research that took what could have been considered uninformed fear more seriously. One part of this debate that has baffled me is the focus on not publishing this kind of work when that is exactly what we need to do to develop realistic response plans. The irony of it is that I would bet the eternal box of donuts that there was much scarier work being done 10 to 20 years ago to weaponize smallpox that was never published because we were not supposed to be doing it. You and I did not have a need to know. They're not saying don't do the work, just don't publish the results. All the minimal risk with none of the valuable gain. And then points out that he would have fun on the TWiV podcast, which I think would be the case.
Yeah, I mean, I like this perspective because I agree that Asilomar was an important point. And I have to point out, Mike, at the New York Academy, this was poo-pooed by some of the participants on the panel. They said Asilomar was a failure, Recombinant Advisory Committee was a failure. And, I just don't see it that way. And I don't like it when the actual facts are twisted to fit someone's view on this.
I don't understand that either, Vincent. And my question is, and I don't know, if you've gleaned this at all from talking to Ron or Yoshi, one of the ideas behind this moratorium on continuing this work was to give the researchers in that field the opportunity to really get together and discuss, where are we going with this? How are we going to do it? To avoid running into this situation in the future. And I'm just wondering whether those discussions are actually occurring or if there's a bit of a lost opportunity here. Because I think it would be much better if the researchers came up with a proposal than something draconian gets laid on top of them from the government or whoever else decides to do it.
I have not heard of any such discussions. I mean, I assume that was what would be happening during the moratorium, but it was over before, as far as I can tell, anything had happened.
Well, my understanding is that the moratorium is unofficially still in effect.
Right. Well,
at least that's what it said in one of those Science or Nature things.
I saw that. But in Dublin, Ron Fouché said it's over yesterday. So, I don't know.
Right. But from statements that folks at NIH have been making, I gather that there is work at that level. And so when you mentioned draconian measures being imposed by the government, it depends on which part of the government is imposing them, how draconian they're going to be.
Sure.
I don't think that this is something that's that's going to be decided legislatively. I think this is something that's going to be decided probably at NIH and at HHS on a broader scale.
I can only hope so.
Yeah.
This document that we were talking about earlier, the United States Government Policy for Oversight of Life Sciences Dual Use Research of Concern, that came out as a result of all this, is the first step, I think, in that. And hopefully that will be part of a larger effort to address dual use in biological science, and hopefully it will recruit a lot of scientists into the discussion.
I think it's probably safe to say that once an organization like HHS gets started on something like this, particularly given the hubbub that surrounded this particular set of publications, this is going to lead to a more fleshed out type of document.
Mm-hmm.
Right. Yeah, I think that's an important outcome of this whole discussion, is that it's really brought it to the forefront where hopefully it will get more attention.
Right. This was something that was supposed to have been done a few years ago, and now that we've been caught with our pants down, we're finally getting around to it.
Right.
Like I said, we made our original recommendations in 2007. So.
Right.
All right, we're going to do some picks, Michael. Do you want to stick around for a bit?
Sure, I'm happy to.
You're happy we had enough of this TWiV business. All right, Rich, what do you have for us this week?
I'm into classics. I have a classic. All right. Brave New World by Aldous Huxley.
Ah, yes.
Probably, a large percentage of the listeners already know about this, but there may be some who don't. And for those who don't, go read it. This is a classic science fiction. I would call it Black Utopia. And in fact, Aldous Huxley apparently wrote this in response to, among other things, inspired by some of the utopian novels that had been written, where he said, well, it might not work out really well. And so this was written in the 30s, and it's about the world in something, it's the 26th century, where he envisions there is no natural human reproduction. All babies are test tube babies entirely, and they are actually reared in the test tube to have different capabilities, and then afterwards conditioned by sleep training to have certain values, so they can all be organized into castes to do different jobs and to be perfectly happy with their place in society, all right? And then, of course, as all of these novels go, there are a couple of misfits that show up who say, wait a minute, and question this whole thing. And it's a fantastic novel.
Excellent.
It keeps coming to mind for me when we talk about the future of genetics in our society, because this is one possible vision. This is the scary vision of where the ability to manipulate biology could go.
Yeah, it's really been a touchstone in all discussions of biology and molecular biology since.
Oh, Alan's gone.
Oh, we lost him again.
Since when?
Are you there?
Oh, am I still here?
You're back. You got cut off when you said since.
Oh, yes, since the book came out.
Right.
Did you just recently reread it, Rich?
No, but I think I should. But it's there on my mind all the time. It was one of my favorites in high school and college. I even wrote an essay about it at one point. But it'd be fun to reread.
All right. You got to pull out that essay and we'll publish it online.
I can't find it. I lost it somewhere along the line.
It's all that stuff. Yeah, it's gone, that paper.
When I retire, I'm going through my whole house. Maybe I'll find it.
All right. Thanks for that. Alan, what do you have?
I have just a little thing on a neat cartography project where somebody went back to 17th century or 18th century ship logs from the Age of Empires and plotted them on a modern map. So these are the trade routes of the British and Dutch and Spanish empires, and they've done this whole mapping of them, and you can see how the different great European powers, how they differed in their shipping of cargo. The Spanish one is almost entirely going to and from South America, whereas the British one is much more spread out. And it's just a really neat graphical representation of a century of history that largely gave us the modern world.
Oh, the Pacific.
This is very cool. The animation is really cool too. They put all of them together and animate them. And one of the things that I notice is that you don't go around the horn unless you have to.
Yeah.
Oh, yeah.
and that it's seasonal.
Yeah. Oh, yeah. It's seasonal, and because of the locations of the trade winds, because of the geographical fact of the trade winds, that determined the settlement patterns for most of these empires.
Yeah, sure.
Interesting.
Pacific is pretty random, isn't it?
Yeah, there wasn't a lot of... You've got James Cook's voyage almost all out there by its lonesome, or his couple of voyages, and not a whole lot else going on because it didn't have... it didn't have the kinds of resources that the 18th century empires wanted.
Yeah, right. This is really cool. Nice. Thank you, Alan. Yeah. My pick is an article I came across this week in the Telegraph. It is all about the Air France Flight 447. It's called Air France Flight 447. Damn it, we're going to crash. So some of you may remember that way back in 1929, a flight from Rio to Paris just disappeared over the ocean. And not too long ago, they found the black box at the bottom of the ocean, and this is a reconstruction of the flight by two authors who essentially, in the analysis of that black box, essentially shows that one of the pilots kind of freaked out in this flight. So they ran into a thunderstorm, the pitot tube froze over, and then the autopilot automatically shut off, and one of the pilots had a bad reaction and just kept trying to get the airplane to climb, and it stalled, and by the time the other two pilots figured out what was wrong, it was in the ocean. So it's a great article going through this, but it also talks about the design of this plane and how it might have contributed to this disaster. So it's science in that you can see that it's all about airplane design.
Yeah, I thought this was really cool.
It really was just an interesting take on this tragic disaster.
Yeah, it was really tragic. They have some of the dialogue towards the end.
Yeah.
Damn it, we're going to crash. This can't be happening.
Oh, the most interesting thing is this notion that plane flies with a joystick.
Right.
Okay. Rather than a big control rig right in front of you. And so, and the control stick returns to neutral after you do any particular maneuver. So if one guy's flying the plane, the other guy's controls don't necessarily, it's not obvious what he's doing.
Right.
Right. So this guy was trying to make the plane climb, and it wasn't immediately obvious to the other people in the cockpit that that's what he was trying to do. Very interesting.
Apparently they don't have an indicator of attitude of some sort, so they couldn't tell. And It says 45 seconds before impact, this guy blurted out that he'd been trying to climb throughout the whole emergency, giving his colleagues the first indication of what had been going wrong.
Yep.
No, don't climb. No. Give me the controls. It's really, really scary. I read it one night. Oh, man. I don't want to fly anymore. Anyway, it's really good writing, though.
Yeah, it's very good.
We have a listener pick of the week from Josh. Dear TWiV doctors, I have a suggestion for a listener pick of the week. Dr. Paul Offit's course on vaccines from UPenn on Coursera. It starts in June, and it is free. It does not appear to be a technical course, but rather a general examination of the history, science, and use of vaccines. As such, it might be of more interest to lay people than people in the field. My assessment might be inaccurate, however, since I have not taken it yet. So there you go. You can take Dr. Offit's course.
Probably very good.
I'm sure. He's a good speaker, good writer.
And knows a heck of a lot about vaccines.
He does. And he's written a couple of books that we have talked about here on TWiV. And he's developed one, the rotavirus vaccine. And you, in fact, Alan, let's see. You did an interview with Maurice Hilleman, and somehow you did you interview also Offit at the same time?
I don't know if I quoted Offit in that. He was there. I did talk to him.
All right, that is TWiV 182. Hope you liked it. It's at twiv.tv on iTunes and at microbeworld.org. And if you have questions or comments, send them in, twiv@twiv.tv. We'll do the Fouchier paper as soon as it comes out. See if it's any different, anything similar, and maybe we'll ask Mike to explain that one too. Mike Imperiale is at the University of Michigan. Thanks for joining us, Mike.
My pleasure, guys.
Really appreciate it.
Great having you.
Thanks very much, Mike. It was great.
I know
You're welcome.
We have had some discussions online, which have been a bit testy at times, and I think it really helps to have you here and have you talk about it in a civilized way.
Well, we're doing what Peter's asking, right?
That's right. That's right. Yeah, we'll have Peter on at some point. Rich Condit, University of Florida, Gainesville.
Gainesville, where it is 89 degrees Fahrenheit and partly sunny, partly cloudy. It's beautiful weather here.
89.
Yep.
Nice.
Great to be here. It's always a good time.
Thank you. And Alan Dove is in Western Massachusetts at alandove.com. Thank you, Alan.
Thank you. It's always a pleasure.
Here in New York City, which is where I am, it is 21 degrees centigrade or Celsius, on its way to 26, apparently. And you can figure out what that is. I know exactly what.
I'm looking it up. I'm looking it up on my iPhone.
Don't translate. Just feel it, man. Just feel it.
We'll probably only hit 22 here in Massachusetts.
And you can find me, by the way. I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.